So I will talk today about the dopaminergic neuron subtypes and their importance for the development of therapies for Parkinson's disease. Dopaminergic neurons have been classically divided by their anatomical location. So in the ventral midbrain region, we have three different locations: the retrorubral field, the ventral tegmental area, and the substantia nigra pars compacta. The substantia nigra pars compacta can be further subdivided in dorsal and ventral. And the ventral neurons in the substantia nigra pars compacta are the ones that project to the striatum, and those are the ones that are involved in Parkinson's disease, so the ones that degenerate. At a molecular level, there are 9 different types of dopaminergic neurons that have been described in mouse, but in human and non-human primates, there have been described 10 neurons, so there is one more type whose function is not really known. But so is it also with many of the other cell types that are now molecularly described. The function is not clear. Now, in this meeting, what we have presented is the characterization of dopaminergic neurons, both in development and in the adult. This is in the human brain. In development, we have analyzed a total of 250,000 cells by single-cell RNA sequencing, and we have identified multiple cell clusters. Now, what we see is that there are a group of progenitor cells, and these progenitors generate progressively a cascade of cells that lead to the formation of dopaminergic neurons. And we see that dopaminergic neurons come in 3 flavors. There are 3 different classes or categories of dopaminergic cells. And within these 3 classes, we have 20 different molecular types or states of dopaminergic neurons. And it is difficult for us to understand whether this is a cell type or a state because we are seeing at a sequence of events. Now, in the adult, this is a little bit more simple because we have a static picture. We have analyzed the midbrain region in patients that have been analyzed by single nuclei RNA sequencing. Out of a total of 3.3 million, we have taken cells. We have only taken the midbrain region and analyzed them for their dopaminergic cells content. We have seen that there are a total of 14 different subtypes of dopaminergic neurons, that is 4 more cells than have been described before. And these cells belong to 3 different classes of dopaminergic neurons. The ones that express SOC6, that are most abundant in the substantia nigra pars compacta, in the ventral part. The cells that express calbindin, that are present in the entire midbrain region. And we have found, for instance, some cells that are new, that are present in the periaqueductal gray. And this is interesting because this region is involved in pain regulation. And in Parkinson's disease, one of the characteristics is that in the non-motor symptoms is the appearance of pain. There is a third class that is also very interesting because it is a mixture of dopaminergic and GABAergic phenotypes. And that indicates that there is a gradient of identities from dopaminergic to GABAergic cells, and this represents an intermediate state that hasn't been described before. Now, all of this indicates that the midbrain region, both in development and in the adult, contains a much more richer molecular diversity of cell types that has been previously described. Now, why this is important? So this is important because until now there has been only one subtype of dopaminergic neuron that has been described to be involved in Parkinson's disease. But if there are 14 cells, so there are more cells, and many of them have not yet been investigated. So there is a chance that some of these other cells may also be involved in disease, and we do not know yet. So this is going to be something that will be an intense area of investigation in the future. Our results also have important implications because we describe all the pathway from very early in mature cells to generate dopaminergic cells. And this information is key in order to develop, for instance, better disease models for Parkinson's disease. We can take an IPS and guide it through development and generate a specific cell subtype of dopaminergic neuron. We can do the same with embryonic stem cells, use this information, the sequence of events, and generate dopaminergic neurons of a specific subtype that can be used, for instance, for cell replacement therapy for transplantation in patients. And we can also use the information by looking at the transcription factors that are important to make a specific type and use them for reprogramming other cell types in the brain and change their identity and then become a specific subtype of dopaminergic neuron. A third implication is that if we understand the adult dopaminergic cell subtypes and we know how do they look like, we now can look at the stem cells that we're using to generate dopaminergic cells and see whether they are identical to the endogenous cells. We can also do the same with iPS cells that we have used to model disease. So by doing that, we have now the standards and we can determine the quality of the cells that we're using both for developing drugs or as a drug in cell transplantation. So this may have important implications for the development of more personalized medicine, because we can develop better cell therapies. We can also develop better models and better drugs for Parkinson's disease patients.